This diagram at first glance is quite scary. It's a family tree of the different types of white blood cells that will be involved in our immune response, and we will study these different families throughout the rest of the week. I think it's useful to have as a reference point because we will quite often see different cellular names and we can get confused as to which part of the immune response or which part of the white blood cell family we are talking about. So today's lesson is focusing on phagocytes. So if we think about the white blood cells that are involved in our immune response, we can kind of split them into two key families. We can split them into the phagocyte family that will all carry out a response in a very similar way, and we can split them into the lymphocyte family. Today we're going to focus on the phagocyte family. Now there are three names of specific types of phagocytes that you will encounter, and they broadly act in a similar way. We can split our phagocyte family into two branches, the neutrophil branch and the macrophage branch. And the only reason I've put the monocytes on there is because if you see the word monocyte, I want you to know that it's basically referring to macrophages. So we're going to look at phagocytes and think about their structure. So phagocytes are a type of white blood cell whose role is to engulf foreign particles and pathogens that have got past the body barriers. Please do not say they eat the pathogens. Do not call them cell-eating cells. That's a GCSE phrase that would not be accepted at A level. And if you were to cut open the long bones in your body, so the femur, for example, is a very long bone, the bone that goes from your hips down to your knee. If we were to cut open the bones and look inside, we would find lots of jelly or bone marrow. And it's in the bone marrow that our stem cells are produced that are able to differentiate into either neutrophils or macrophages. They'll differentiate into neutrophils or monocytes, and then the monocytes is the name of this phagocyte that travels in the blood, and once it gets to the tissues, it's then referred to as a macrophage. And if we were to actually compare the two different types of phagocytes, the neutrophil family are very easy to identify under a microscope because you can see here their nucleus has a very strange shape. It's described as being a trilobed or multilobed nucleus. So it doesn't just appear as a single blob in the middle of the cell like we're used to seeing. It's got many lobes surrounded by the nuclear envelope. They make up the really large proportion of the total white blood cell family, and they are quite a small cell because they've got to be able to squeeze into tissues to be able to carry out phagocytosis in tissues. And because it's the first non-specific immune response cell that's involved, they tend to be short-lived. Their numbers obviously will increase during an active infection. And when we looked at tissue fluid formation earlier on in the course, we talked about the fact that in our capillaries, we have these small pores between the cells that line the capillary wall. And those pores are big enough to allow the neutrophils to be able to squeeze out of the capillaries and into the tissue fluid. And by having this trilobed nucleus and relatively small size, that allows them to squeeze through the gaps. But their function is to fully engulf and fully digest a pathogen, and we'll look at how that happens on the next slide. Our macrophage family of phagocytes tend to be slightly larger, and they're predominantly found in the organs and carry out phagocytosis in the cells within an organ. They tend to be slightly longer lived, and then they have a role in initiating or starting a specific immune response. And they act in a very similar way to neutrophils, but the final stage is slightly different. So rather than fully digesting the pathogens like would happen with the neutrophils, they will cut up the pathogens and they will display the pathogen antigens on their own particular surface to generate a cell called an antigen presenting cell. If we were to look at both of these types of cells under a microscope, the cytoplasm would be described as having a granular appearance because there are lots and lots of lysosomes containing hydrolytic enzymes within the cytoplasm of our neutrophils and our macrophages that have a role in containing hydrolytic enzymes that will allow us to cut up the pathogens or to fully digest the pathogens.